Hello everyone. In this video, we are going to start with Unit 5 Enzymes. We are aware that in the body of all living organisms like animals, plants, or microscopic organisms like amoeba, bacteria, etc., there are many chemical reactions which take place in every few seconds or in every few minutes. Now, the rate of these chemical reactions in the cells of these living organisms is supposed to be maintained in the similar ways, because if the rate of reaction becomes very slow or if the rate of reaction becomes very high, what happens is the cell will not be able to function in the appropriate way. Okay, and sometimes it might lead to the death of these cells, especially in the unicellular organisms. Okay, we are aware that in our surrounding, in our day-to-day life, we know that there are many industries, chemical industries are present around us, and in these chemical industries also many chemical reactions takes place. Now there also the rate of these chemical reactions are supposed to be maintained. What happens is there actually a substance is used in order to maintain or in order to increase the rate of reaction, and these substances are known as catalyst. Okay, so what these substances are called? These substances are known as catalyst. So catalysts are the substances that can increase the rate of chemical reactions without being used up or undergoing any change. Now this point is very important whenever you write the definition of catalyst, that whenever a catalyst is used in a chemical reaction, this catalyst is not being used up. Okay, after the reaction gets completed, the catalyst is not being used up and it do not undergo any change. And here, instead of using the word increase, we can also use another word, alter. Okay, so catalyst is a substance that can increase the rate of chemical reactions or it can alter the rate of chemical reactions without being used up or undergoing any change. Now, I've told you a little before that many chemical reactions occur in the body of living organisms also. So in the body of living organisms also, there should be a catalyst-like substance, okay, that can actually increase the rate of these chemical reactions in the body of living organisms. Okay, so these naturally occurring catalysts in the body of living organisms are known as enzymes. Fine, so these naturally occurring catalysts which occur or which are present in the body of living organisms are known as enzymes. So these enzymes are biocatalysts, very important. These enzymes are biocatalysts. We are calling it biocatalyst because the meaning of word bio refers to living organisms. Okay, so the meaning of word bio refers to living organisms or living cells. And catalyst means the substances which can increase the rate of reaction or substances which can alter the rate of these chemical reactions, rate of these metabolic reactions taking place in living cells, okay, without undergoing any kind of change. So enzymes are biocatalysts. That means they control the metabolic reactions taking place in living cells without undergoing any change, okay. And very important point, this question is asked many times in IGCSE papers, especially in paper 1 in your MCQs, that enzymes are made up of which biomolecule. Enzyme itself is a biomolecule, but it's made up of which category of biomolecule. Okay, so enzymes are made up of proteins. Sometimes in exam, actually the definition of enzyme is asked for 3 marks. Now, whenever the definition of enzyme is asked for 3 marks, you can write down following 2 points. Either you can write down that enzymes are biocatalysts and they are made up of protein. So if you write down that enzymes are biocatalysts and they are made up of proteins, you will get 3 marks. Why? Because when you write down this one particular term, biocatalyst, okay, so this term shows that these enzymes are present in living organisms, as we are mentioning here bio, and these enzymes are catalysts. That means they increase the rate of metabolic reactions taking place in living cells without undergoing any kind of change. Okay, so just for writing this one particular term biocatalyst, you can get 2 marks, and if you also write down they are made up of proteins, you will get 3 marks. But remember, the mistakes which are done generally by the students is when you write that enzymes are biocatalysts, and then you write out third point that they control all the metabolic reactions taking place in living cells without undergoing any change. That's it. So you won't be awarded 3 marks for this. You'll get only 2 marks. Why? Because the second sentence is actually the elaborative form of the word biocatalyst. Okay. So when you write the term biocatalyst, that means you are mentioning that enzymes control all the metabolic reactions taking place in living cells without undergoing any kind of change. So it's must to mention that enzymes are made up of proteins. Fine. And what is the importance of enzyme? Enzymes are important because they ensure that rate of metabolic reactions is always high in living cells. This is very important. Rate of metabolic reactions is always high in living cells to sustain life. Okay. Now, for example, if these chemical reactions or metabolic reactions which are occurring in living cells, if they will take place at very slow rate, so what will happen? The cell will function very slowly. Now, if the cell function will be very slow, definitely the organism's body or organs function will be very slow. Hence, it will be difficult to sustain life. Okay, it will be difficult for that particular organism to stay alive. Okay, so in order to maintain the speed of these chemical reactions, in order to keep all these metabolic reactions always high in living cells, enzymes are used. Okay, so without enzymes, what happens? The speed of these reactions will decrease. Okay, so without enzymes, what happens? The speed of this reaction will decrease. Okay, so this is the importance of enzyme. Next, we will look into the examples of enzyme. The first example is carbohydrates. Now, as the name suggests here, this is carbohydrates. Okay, so carbohydrates means the enzyme which digests carbohydrates. Okay, the enzyme which helps in digestion of carbohydrates. Example: salivary amylase. Salivary amylase is present in our saliva. This is the enzyme which digests the starch present in the food. So whenever you start eating, like chapati, or whenever you take rice, this chapati or rice is having starch in it. Okay, so the starch, the food gets mixed with the saliva. In saliva, enzyme salivary amylase is present, which will digest starch into maltose. Okay, so this salivary amylase digests starch into maltose. Okay. Next example of enzyme is protease. Now, protease, the name suggests it digests proteins. So there are actually different examples of protease. There are different examples of carbohydrates. Okay. So this protease digests proteins into amino acids. So what is the function of protease? Protease enzyme digests proteins into amino acids. Third enzyme is lipase. Okay. So the function of lipase is to digest fat. Lipase digests fat into fatty acids and glycerol. Fine. So lipase will digest fat into fatty acids and glycerol. The next enzyme is catalase. Now this catalase enzyme is very important. Also important as per the exam point of view. Questions are asked on this catalase enzyme many times. Okay. This catalase enzyme is present in all the living cells. Okay, it is actually, I've written here present in animal liver cells, but actually the correct thing is it is present in all cells. Okay, it is present in all the cells, whether it is plants, animals, amoeba, bacteria, prokaryote, eukaryote, any type of cell is having the enzyme catalase. Now the function of this enzyme catalase is whenever metabolic reactions takes place in all the living cells, a highly poisonous, or you can say a highly toxic byproduct is produced as a result of these metabolic reactions, and that byproduct is hydrogen peroxide. Okay, the byproduct is hydrogen peroxide. Now the moment this toxic byproduct is produced after metabolic reaction in cells, immediately this byproduct is supposed to be broken down into water. Okay, water and oxygen. Okay, so there are enzymes called catalase enzymes present in all living cells, because the moment hydrogen peroxide is produced as a result of a byproduct in any metabolic reaction. So what happens is catalyst will break down this hydrogen peroxide into water and oxygen. Fine. So you are aware that water and oxygen both are not harmful for our body. Okay, but hydrogen peroxide is toxic. So the function of this catalase enzyme is to break down hydrogen peroxide into water and oxygen in the living cells. Next enzyme is starch phosphorylase. Okay, actually the function of these enzymes is not only to break down large molecules into smaller molecules. Enzymes are also used up in building up of large molecules. Like it takes smaller molecules, it takes monomers, join them all together and forms the large polymer molecule. Okay, so one of the example of such enzyme is starch phosphorylase. Now what this starch phosphorylase does, it is present in plant cells, and this starch phosphorylase converts glucose into starch. Okay, now you know that in the process of photosynthesis glucose is produced. Okay, now the entire amount of glucose which is produced in the process of photosynthesis cannot be used immediately at all together at one time, cannot be used by the plants. So always this excess amount of glucose is stored in the leaves of plant in the form of starch. Now the enzyme which helps in conversion of glucose into starch is starch phosphorylase. So I hope these examples are clear to you. Next, we will understand the mechanism of enzyme action. Very important topic as per the exam point of view again. Okay. Okay. So mechanism of enzyme action. So the hypothesis which actually supports the mechanism of enzyme action is known as lock and key hypothesis. You are aware that you have seen that lock and key at your house. Okay, so the lock is actually acts like an enzyme, and the key present acts like the substrate molecule. Now what are these substrate molecules? Okay, what are products? I'll just let you know from the previous slide. If you look here in the previous slide, here hydrogen peroxide. Okay, so this hydrogen peroxide is actually a substrate molecule. That means in chemistry you have studied about reactants, isn't it? So all your reactants are actually the substrate molecule which take part in the chemical reactions. So in this particular chemical reaction, hydrogen peroxide is a substrate molecule. Okay, catalase is the enzyme. Okay, catalase is the enzyme, and water and oxygen, they are the products. So what happens is, as per lock and key hypothesis, the enzyme acts like a lock. Okay, and every enzyme molecule which is acting like which acts like a lock is having a particular shape. Okay, is actually having a particular dent with specific shape. Now this dent with specific shape in enzyme is known as active site. This is again a very important point. Okay, what this dent in enzyme is known as? This dent in enzyme, and this dent in enzyme is having a very specific shape. Okay, so this dent in enzyme with very specific shape is actually known as active site of the enzyme. Okay, so according to lock and key hypothesis, the enzyme molecule is like a lock. Okay, the enzyme is having active site, and the substrate molecule, that means the reactant molecule which is taking part in the chemical reaction, is having a complementary shape to the enzyme. That means the shape of active site of the enzyme is complementary to the shape of the substrate molecule. Okay, now as the shape of active site of enzyme is complementary to the shape of the substrate molecule, the substrate molecule perfectly fits in the enzyme molecule. Like the key fits into the lock. Okay, similarly the substrate molecule also fits in the active site of the enzyme. Okay. Now you can see here in the picture, the substrate must be a perfect fit. That means the substrate, the shape of the substrate molecule is complementary to the shape of the active site of the enzyme. Hence, it gets perfectly fit into the active site. Okay. And this particular complex of enzyme and substrate is known as enzyme substrate complex. Okay. And once enzyme will, get fit with the substrate molecule, enzyme will start acting on substrate molecule, and it will finally break substrate molecule into the products. Okay, it will break substrate molecules into the products. But look here carefully that the entire enzyme molecule is not undergone any kind of change after during the entire chemical reaction. Okay, so the enzyme molecule comes out as such without being used up or without undergoing any kind of change. Fine. So this is lock and key hypothesis, where enzyme molecule acts like a lock, substrate molecule acts like a key. Okay, the shape of active site of enzyme is complementary to the shape of substrate molecule. Hence, substrate molecule is able to fit into the active site of enzyme perfectly. Okay, now enzyme starts acting on the substrate molecule, and it breaks down substrate molecule into products without undergoing any kind of change or without being used up in the chemical reaction. Next is enzyme has an active site. Okay, I've already told you in the previous slide that every enzyme has an active site. Okay, now in case if the definition of active site is asked, so what you will write, that the active site is the region of the enzyme where substrate molecules bind and undergo a chemical reaction. Okay, so what is active site? Active site is a part of enzyme, or it is a region in the enzyme where substrate molecule binds and undergo a chemical reaction. And why a substrate molecule is able to bind and undergo a chemical reaction? Because the shape of the active site of the enzyme and the shape of the substrate molecule is complementary to the each other. Okay, so this is again important. The shape of active site is complementary to the shape of the substrate molecule, which makes it highly specific. Okay, now again this is another very important point, that the enzyme action is highly specific. Why it is highly specific? Because the shape of the active site of an enzyme is always complementary to the shape of the substrate molecule on which it is supposed to act. Okay, I'll explain this thing a little later. Now look here, enzyme acts like a key lock, substrate molecule acts like a key. When substrate molecule fits into the enzyme, okay, it forms enzyme substrate complex. Now enzyme starts acting on the substrate and it forms product. Plus enzyme comes out of the chemical reaction as such without being used up or without undergoing any kind of change. Next is properties of enzymes. The first very important property of enzyme is, I've told you earlier, that they are made up of proteins. Okay. Second, these enzymes, they work, in the best possible way at a particular temperature, which is called optimum temperature. Okay, so these enzymes are made inactive at high temperature. For example, all the enzymes in our body. Okay, all the enzymes in our body, for example, the internal body temperature is 37 degrees Celsius. So all enzymes in our body, they works best at 37 degrees Celsius, or you can say between 37 to 40 degrees Celsius. Okay, so if the temperature of a body goes above 40 degrees Celsius, okay, so what happens? The enzymes will become inactive. Okay, instead of saying inactive, we can say the enzymes get completely damaged. The enzymes get damaged completely at high temperature. Okay, that is damaged by heat. So all these enzymes, they work best at a particular temperature, which is called optimum temperature. And the example given is, all enzymes in human body works best at 37 degrees Celsius. Fourth example is, all these enzymes, they work best at a particular pH. Okay, so example of this pH is pepsin in stomach. You have studied in your lower classes that in our stomach, gastric juice is released. This juice contains an enzyme which is called pepsin. Okay. So this pepsin enzyme helps in digestion of protein. Okay. The function of this pepsin enzyme in stomach is to digest protein into amino acids. Okay. Now this pepsin always works best in acidic condition. Okay. So this pepsin works best in acidic condition. But after the food goes to small intestine, the first part of small intestine, that is duodenum, what happens is there the conditions present is alkaline. Okay. So there another enzymes act on the food which are known as trypsin and lipase. Okay. What they are called? Trypsin and lipase. Okay. So this trypsin and lipase, they always require alkaline condition for their work. Okay. So trypsin and lipase in duodenum works best in alkaline condition. Okay. The next, these enzymes are highly specific in nature. Okay. The meaning of this is highly specific in nature. That means a particular type of enzyme can only act on a particular type of substrate molecule. Okay. It cannot digest all the substrate molecule, or it cannot digest any random substrate molecule which with which it collides. Okay. So for example, if you take the enzyme catalase, I've told you recently before few minutes about catalase enzyme present in all living cells. Catalase enzyme breaks down the toxic byproduct of metabolic reactions, that is hydrogen peroxide, into water and oxygen. Okay. So catalase enzyme breaks down hydrogen peroxide into water and oxygen. Now what happens is, this catalase enzyme is having the active site, and the shape of active site of catalase enzyme is complementary to the shape of hydrogen peroxide molecule. Okay. No other reactant molecule or no other any XYZ substrate molecule can just come and fit into the active site of catalase molecule. That's not possible. Okay. That's the reason we say that enzymes are highly specific in nature, because one type of enzyme can only act on one type of substrate molecule. Okay. It cannot form enzyme substrate complex with any XYZ substrate molecule. Next, enzymes are biocatalysts. Okay, so enzymes are made up of protein and they are biocatalysts. These 3 points you can include in the definition of enzyme. So we are done with this particular topic. In next video, we will discuss about factors affecting enzyme actions. Okay.